So this is genome scale evaluation of microbial metabolism for feasibility in industrial chemical production. I am JB King.
I'm Isaac Patesnick.
And I'm Mason Rasmussen.
So regarding the project relevance, large-scale production of various chemical products using microorganisms has been the subject of much research and development in recent decades. While traditional methods of chemical manufacturing are well studied and have improved in their efficiency over time, there are still drawbacks that make microorganism production a worthwhile endeavor. One of the most impactful advantages of using microorganisms is their sustainability. While traditional methods of chemical production can have high energy usage, greenhouse gas emissions, and toxic byproducts, microbial processes typically operate under conditions that minimize these disadvantages. Today, approximately 90% of chemicals are generated from fossil fuels as carbon and energy sources. Also, substitution of petrochemicals with bio-based alternatives could reduce fuel-associated greenhouse emissions by 75 to 80%. New advances like genome editing, adaptive laboratory evolution, computational protein folding software, and library screening technologies have all made bioproduction more feasible. Although there are many benefits to using microorganisms in industrial settings, the main barrier is economic feasibility. It is therefore necessary to achieve high yields of products in the operation of these plants. Another advantage of using microorganisms is the ability to produce products that are difficult to synthesize chemically, like amino acids, vitamins, biopolymers, and biofuels.
In terms of the goals of a project, we intend to model organism growth and determine which organisms are viable in producing substances in an industrial environment. We also want to evaluate microbe metabolisms under varying media to get the best return on investment, and then discuss challenges and limitations regarding the usages of microbes.
Looking at our models and our methods that we used, first, in order to find our models, we used the BIGG models database and searched for our individual microbes and picked the models that we found met our needs the best. This oftentimes meant picking the most recent model, as we figured it was the most current and up-to-date model for each microbe. In order to actually use the models that we selected and look at their metabolic function, we utilized constraint-based reconstruction and analysis, or COBRA Toolbox, which used the Matlab software suite. Within the COBRA Toolbox, we were then able to model, analyze, and predict the metabolic phenotypes of each microbe. In our research, we then used the capabilities of the COBRA Toolbox itself to reconstruct these genome scale metabolic networks for each microorganism, where we were then able to conduct our research by analyzing various chemical pathways of interest.
So for the first organism that we looked at, it was Clostridium ngali. Clostridium are gram-positive anaerobic bacteria that are commonly found in soil and marine sediments. They have been used historically in the commercial production of several solvents, including acetone, butanol, and ethanol. The process in which they make the acetone, butanol, and ethanol is known as ABE fermentation, and this process was developed by Chaim Weizmann and prior to reliance on petrochemicals, this was the main way that we got acetone and butanol and ethanol. Well, got acetone and butanol. For Clostridium, the reactions of interest were the acetone and ethanol production. Shown here is the metabolic framework of the Clostridium model that we're using, which was developed by Sahin et al. In 2013. The model also goes by the title of IHN 637, and I looked at constraining Clostridium to glucose feed. Whenever it came to the results in terms of the acetone production, it was found that Clostridium produced acetone much better than E. coli, which was the reference microbe listed in a paper related to this. So in an industrial setting, if we were wanting to make acetone using a microbe, Clostridium would be the most viable option. But whenever it came to the next reaction of interest in terms of ethanol production, Clostridium did not do so well when comparing ethanol production in Clostridium to that of yeast, which is where we get most of our ethanol from today. So if we were to develop an ethanol plant, we would want to go with yeast as opposed to Clostridium.
So our next organism of interest was Saccharomyces cerevisiae, also known as yeast. Yeast is a unicellular fungus that's widely used in industrial applications. Its entire genome has been sequenced and contains around 6,000 genes. Due to its unique metabolism, yeast is considered a model organism. One aspect of its metabolism that makes it unique is the Crabtree effect, which allows it to produce ethanol and other two-carbon compounds from pyruvate even when oxygen is present, instead of using aerobic respiration, which is more efficient. When it accumulates ethanol, this is toxic to most other microbial organisms, so it effectively eliminates the competition. Another reason yeast is widely used in industry is its tolerance to high sugar concentrations. So as stated, the main industrial application of yeast is the production of ethanol under anaerobic conditions. It uses glycolysis to catabolize sugars into pyruvate, which is then converted to acetaldehyde and carbon dioxide by pyruvate decarboxylase, and finally reduced into ethanol by alcohol dehydrogenase. It is also possible to produce higher alcohols, such as propanol and butanol, from genetically modified strains of yeast. The characteristics that make yeast a good ethanogen include its high ethanol production, stress tolerance, genetic flexibility, and scalability. To optimize alcohol production, the right nutrients need to be supplied, including fermentable carbohydrates, nitrogen, vitamins, oxygen, and minerals. The temperature of the fermentation should be monitored during the whole process to prevent the biosynthesis of stress response factors like heat shock proteins and trehalose. If starch or sugar are used as feedstocks, there can be overproduction of glycerol, which causes osmotic stress that decreases alcohol fermentation. Now, looking at the results, this figure on the left shows the biomass growth rate at varying glucose and oxygen uptake rates. As you can see, there's no biomass growth when the oxygen uptake rate is 0, and that at high uptake rates of both glucose and oxygen, the biomass growth rate increases. And then the figure on the right shows the ethanol secretion at varying glucose and oxygen uptake rates. We can see that increasing ethanol rates occur at lower oxygen concentrations and higher glucose concentrations, with the best being at the highest at 0 oxygen uptake. So in industrial applications, you need to find a balance between the biomass growth rate and the ethanol production. And then this figure on the top shows the ethanol production versus the ammonium uptake at growth optimal scenario and in an ethanol optimal scenario. We can see that there's higher production of ethanol at the ethanol optimal scenario, because in the growth optimal scenario, the nutrients are redirected towards the formation of new biomass and redirected away from ethanol production. And then this figure at the bottom shows biomass growth rate at varying ammonium and oxygen uptakes. We can see that the biomass growth rate increases at higher oxygen and higher ammonium uptake rates, with the limit and the ammonium uptake rate, after which you would have decreasing yields. And then there's also no biomass growth rate when there's 0 oxygen and 0 ammonia.
Looking at our final microbe, I decided to look at Corynebacterium glutamicum. This is an industrial producer of amino acids specifically glutamine and lysine. Specifically for my study, I chose to look at lysine production. In C. glutamicum, lysine is growth coupled, meaning that under normal conditions, all lysine produced is going to be used in growth. And if growth is not occurring, then lysine production is either going to stop or lysine will accumulate within the cytoplasm. As such, production is very highly regulated, as it's both energy intensive and highly resource and highly expensive in terms of resources. And so lysine secretion does not naturally occur. To fix this, though, in industrial settings, producers utilize a fed batch feed strategy and induced nitrogen limitation by the media. During batch phase, growth is allowed, which causes an accumulation of cells leading to very high density, leading to high cell density. And then during the fed phase, lysine is able to be produced. In limiting the nitrogen, growth is slowed while carbon sources are left high, leading to metabolic pathways for lysine production still being open, allowing for greater lysine production. Specifically, when looking at lysine production, the max you will often see is when growth is at a minimum, or vice versa. When growth is at a maximum, lysine production is going to be at a minimum. The model I decided to use was ICGB 21FR, which was the most current model I could find for C. glutamicum, made in 2017 and since updated. Within the model, the main reaction for synthesizing lysine was the DAP-DC reaction, which is shown here, involving the cleavage of diaminopimalaic acid and produces L-lysine. This reaction occurred within the cytoplasm, and since lysine is not normally secreted, for my model specifically, I needed to add an extra reaction where lysine was able to be exported from the cytoplasm into the periplasm, so then it could be further exported into the extracellular region and then secreted, which allowed me to model my lysine production. Moving on to the results of my research, the figure on the left shows the max yield of lysine per carbon source on a few different medias, those being carbon-rich, nitrogen-limited, oxygen-rich, and normal, which is just standard conditions. Here we can see that for almost every media, sucrose showed the highest lysine yield compared to fructose and glucose. Glucose being the top row, fructose being the middle row, and sucrose being the bottom row. Now, admittedly, these yields are far too high, as common literature values range from 0.5 to 0.7 grams of lysine per gram of carbon source. However, it is worth noting that simply relatively speaking, the sucrose yield is higher, which could have implications when looking at feedstock options. Moving on, the figure on the right shows the trade-off of lysine yield per carbon source and the growth rate on a nitrogen-limited media. The nitrogen-limited media specifically was chosen as it's the most common growth media in industrial settings. And so I determined that it was the most appropriate to evaluate growth and lysine yield on. In the plot, we also see that sucrose has the highest max lysine production and highest max growth rate, followed by fructose and then glucose. These values are going to be slightly different from the table seen on the left, as the figures are operating on different model objectives. The model on the left is optimizing only for lysine production, while the model on the right optimizes both lysine production and growth. And so that changes the pathways being utilized in each case, changing the production and yield in both instances. Again, however, the values seen in the right plot are higher than current literature values, but the important thing is the relative difference that's seen, mainly because glucose is the most common carbon source used in industry right now. While glucose is cheaper than refined sucrose, molasses, which is a combination of sucrose and fructose, is a far cheaper option than both refined sucrose and glucose. This would mean that the potential for using molasses as opposed to glucose as a feedstock could lead to better economic outputs in lysine production. However, we do know from literature sources that sucrose and fructose lead to more complex metabolisms within the microbe, which are surely not accounted for in the model, as it only has roughly 1,200 reactions within it. And so the findings may be a little exaggerated. However, this does mean that if the metabolism was engineered to do so, C-glutamicum could have higher lysine yields using molasses compared to glucose, which would significantly cut the cost of feedstock. Overall, we found that Clostridium has shown to be a much better producer of acetone than an ethanol producer when looking at it in industrial settings. For yeast, we found that decreasing oxygen availability greatly increases ethanol production, yet causes drops in the growth rate. And finally, for C-glutamicum, we found that alternatives to current glucose feedstocks like fructose and sucrose may be viable options going forward. Looking at the limitations of our study, the main issue that we found was that we were subject to the model itself, the models themselves, in which case we had to rely on only existing models, which were either wrong or too small to give accurate results. Another limitation kind of related to that is that most GEMs do not look at enzyme kinetics or protein abundance, which may be leading to the improbable fluxes in our models, as many of the rate limiting steps at the protein level at least aren't being accounted for. And then finally, there may have been a lack of regulation within the models, as unless you specifically defined bounds, the uptake or regulation of certain pathways was not accounted for, which could have led to unlimited fluxes within the models, therefore leading to the high fluxes that we saw.